Listo. Hola, buenos días. Estamos transmitiendo la segunda sesión del seminario que hacemos junto con investigadores de Francia en este proyecto que tenemos en común. Esperamos que se queden con nosotros. Hay 2 pláticas hoy, a las 10 y a las 11, y mañana otras 2. Y para presentar a nuestra ponente de hoy está Pierre-Henri, que nos va a platicar un poco de ella.
Hola a todos y todos. Voy a cambiar de lenguaje porque va a ser más fácil para mí. I will introduce you Marie, who is a PhD student working with Yannick, who you can see also on the screen, and me. She will explain you what she's been working on over the last 3 years and some months about the role of cholesterol in fine-tuning the T-cell early activation. I hope you will find this work as interesting as we do, and that will at least open some connections with some of the interest of the lab here about the problems of liver, the problem of fatty liver, the problems that will link this kind of molecules to therapies, but also diseases. Marie, you can go.
Okay, thanks for the introduction. So, as Pierre-Henri said, I'm a PhD student between the Center of Immunology in Marseille and the Laboratoire Adhésion Inflammation. And we'll start talking about fats, dyslipidemia, and a bit of inflammation. So, yes. First, I have to say people with obesity undergo systemic low-grade inflammation coupled to more aggressive adipose tissue inflammation. And on the other hand, dyslipidemia occurs frequently in autoimmune diseases, and an unbalanced lipid metabolism contributes to accelerate inflammatory response. So this raises a first link between dyslipidemia and inflammation. And if we want to be a bit more precise, we have collaborators in Paris who show that in dyslipidemia patients wearing mutations in the cholesterol exporters ABCA1 and ABCG1, these patients exhibit a shift in the level of inflammatory cytokines in their serums. And these cytokines are produced by T cells. But first, maybe a small definition point. So inflammation is initiated when the immune systems detect the presence of potentially harmful stimuli, such as pathogens or damaged cells. And this initiates leukocyte recruitment. Among them, T cells. So T cells are responsible for the cell-mediated adaptive immune response. Some T cells, the CD4 plus T cells, activate and help in activating macrophages, help in activating B cells, and in regulating the immune response. And some other T cells will be responsible for killing cells infected by viruses or cancer cells. These are the CD8 plus T cells. And to be efficient, these T cells need to activate. And this occurs when an antigen-presenting cells bind to the T cell receptors. But I'll come back to that later. So together, this link between dyslipidemia, inflammation, and T cells led people to investigate the regulations of lipids within T cells. And they showed that lipids, and for instance, cholesterol, play a major role in T cell homeostasis, and that mutations in cholesterol processing proteins affect T cell biology. But we still don't really understand how cholesterol tunes T cell homeostasis at the molecular level. And more precisely, molecular regulations of T cell activation by cholesterol remains unclear. And that's what I'm looking at during my PhD, meaning how cholesterol and lipids fine-tune T cell early activation. So the activation of T cells starts with the engagements of a really special receptors, the T cell receptors. So what makes it so special? So here are naive T cells waiting in the spleen and in the lymph nodes to activate. So these naive T cells will bear plenty of T cell receptors with the TCR chains surrounded by the CD3 chains. These T cells is waiting to meet the good antigen-presenting cells that will present the antigen that will fit to the TCR to then build an immune response an immune synapse that will transmit the signals for the T cell activations. So when it looks really easy on that kind of cartoons, in the reality, it looks more a bit like that, a T cell surrounded by thousands of other cells. And these thousands of other cells will establish contacts with the TCR. So the TCR will need to discriminate between non-specific antigens between other molecules, and will need to be able to find the good antigens, really rare signals among everything of that bindings. So to be efficient, the TCR needs to fulfill these kind of properties. It needs to be selective to activate only when it finds the good antigens. It needs to be sensitive to be able to detect these rare signals. It will use the discriminations. It will use as discrimination factors the on rate and the off rates of that bindings. This will also introduce or sense forces on the TCR and use this mechanosensitivity as a discrimination factors. And this will lead to conformational changes that will then finally lead to a quick transmission of the activation signals. So all that properties of the TCR assure that the TCR will activate or will trigger activation signals, assuring that we will have immune response, but will assure that the TCR will not activate when the antigen is not the good one, preventing autoimmune responses. And even if we identify all that properties, a question remains, and this is, how do these naive T cells to translate these rare, diverse signals into a strong, specific, and really, really rapid immune response? And first answers is in the properties that I told you, I mean the conformational changes. So in the TCR, you will find cytoplasmic tails from the CD3 domains. And these cytoplasmic tails will bear basic-rich sequences that are positively charged. And these positively charged basic-rich sequences will interact with the negative phospholipids of the membrane. These induce a close proximity from between the cytoplasmic tails and the membrane. And when an activation signal is produced, some signals like entry of calciums will disturb this interaction between the cytoplasmic tails and the membrane, and will lead to a release of that cytoplasmic tails in the cytoplasm of the T cells. These make items available. These items are motifs on the CD3 that will be phosphorylated by kinases like LCK and then transmit activation signals. And last question is, how does that membrane organizations tune this TCR conformation and conformational switch, and how this does affect the T cell activation? So to study proximal event of activations, we wanted to create perturbations of the lipidic organization of the plasma membrane. In that sense, we chose to target the cholesterol exporters ABCA1 and ABCG1, which are already known to tune the plasma membrane composition. To do so, we in the lab, we created a conditional double knockout of ABCA1 and ABCG1 only in T cells, and looked at, did we indeed impair the cholesterol transport within that cells? So that's the first thing we checked. And to do so, we tracked the binding of philippine over time in cytometry. So philippine is a small component that binds to the free cholesterol of the plasma membrane, and that will only fluoresce when it's bound to cholesterol. And here, using cytometry, you can see that as soon as we put philippines in the medium, we see an increase of the philippine binding to the T cells, especially in the WKO T cells compared to the wild-type T cells. This means that we have indeed in our ABCA1 and ABCG1 WKO T cells an increase in the free cholesterol in the outer leaflet of the plasma membrane. And also lipid storage is increased in our WKO T cells as we can see more and bigger lipid droplets. So this was first properties, but we performed a more precise lipidomic analysis. And we got plenty of results, but the most striking one is that we see that the phospholipids and fatty acids, for instance, monounsaturated fatty acids, like oleic acids, and these type of polyunsaturated fatty acids are used to build neutral lipids and to be stored in, so these kind of lipids are used to be stored as in the form of neutral lipids in the lipid droplets. So this is coherent with what we see using microscopy, but this is also interesting in the sense that that these phospholipids have plenty of properties. For example, it reduced the availability of that phospholipids from membrane reactivity. It reduced these anti-inflammatory lipids, and it's also decreased these fatty acids that are initially available for oxidative phosphorylations and fatty acid oxidations in the mitochondria. Why is it important? It's important because it has been shown that in naive and memory T cells, so in the T cells that are not activated yet, these T cells tend to use more oxidative phosphorylations and fatty acid oxidations. But as soon as the T cells will activate, the metabolism will switch to use glycolysis. So probably at the resting states of our WKO T cells can't use oxidative phosphorylations and fatty acid oxidations as would do wild-type T cells. And when we looked at metabolisms, we see that in our WKO, and for instance, in CD4 WKO, we have less important dependency on mitochondria, but we have a bigger importance of the glycolysis. And indeed, when we looked at the mitochondria analysis, we see that we have less mitochondria, going in the sense of probably a bit, going in the sense of less utilization of fatty acid oxidations and oxidative phosphorylations. And when we activate the T cells, we increase that use of glycolysis, but we still recruit mitochondria and increase the use of mitochondria. So it means that in our WKO T cells, they use at the naive states more glycolysis, which is a metabolic pathway that is used normally in activated T cells, but that we still have mitochondria to support activations. And all of that goes in the sense of what we find doing a proteomic analysis. For example, so we took our T cells and used mass spectroscopy to analyze the protein that we have. And among all the proteins that are shifts in our WKO T cells, we see a huge shifts in the cholesterol biosynthesis, lipids biosynthesis. And we see that the metabolic switch is possible in our WKO T cells, but comes later in the process of activations, probably because they're already able to support glycolysis. So finally, all of that kind of lipid alterations leads to morphological alterations of our WKO T cells. So as I already said, we see more lipid droplets, but also at the level of the membrane, we see alterations in the sense that when in our wild-type T cells, we see more smooth surfaces. In the WKO T cells, we see surfaces with increased density of microvilli. So with all that kind of experiments, it allows us to really characterize the kind of cells we have, the lipids that are available in the WKOT cells, and the lipids alterations that we induced using the WKO. And now that we characterized the cells that we use, we can go a bit deeper into the subject that interest us, meaning activations. And to look at activations, we perform a kind of a multi-scale and multidisciplinary explorations, looking at the T cell populations, the T cell activations, meaning the transmission of the signals, and the molecular mechanisms. So our fatty T cells, first we have fatty T cells, and that it looks that they develop normally, meaning in the thymus, when we look at the cell distributions, we don't see differences between wild type and WKO T cells. But as soon as they arrive in the periphery, meaning in other organs, and for instance in lymph nodes and spleens, we see a loss of T cells. So we have less T cells in the periphery, and we see a switch in the CD4 to CD8 plus ratio. I mean, normally we have kind of the same amount of CD4 and CD8 plus T cells in lymph nodes and spleen. And in our WKO T cells, we see an increased loss of CD8 plus T cells to compared with CD4 plus T cells, meaning that that CD8 plus T cells might have an increased sensibility to the lipid alterations compared to CD4 plus T cells. And then finally, when we looked at the morphology of organs, so this histology study has been performed by Thibaut Janson, and it's kind of really preliminary. But we see disruptions of the general centers of the lymphoid zones within the spleens. So alterations of the lipids in the T cells really alters the organizations and the architectures of the spleens, but this will require more investigation from this. So we have T cells, but we have less T cells and less of CD8 plus T cells. And within that CD8 plus T cells, we see an increase of the expression of CD44, which is an inflammation marker of the CD4 plus on the CD8 plus T cells. So we see less of that naive T cells and more of that central memory and effector memories populations within the WKO CD8 plus T cells. So even if we didn't activate the T cells, we see this increase of T cells that have already undergo some activation signaling. And in the counterpart, we see an increase in the regulatory T cells among the CD4 plus T cells. And together, this increase of central and effector memory T cells and this increase of regulatory T cells is typical from an inflammatory landscape among the CD8 plus T cells. So it seems that even if we only affect the T cells, we push the T cells populations into an inflammatory populations. And if we go even deeper within the populations, we can look at the expression of the TCR again using some staining and cytometry. And what we see is that we always have more TCR, or we always have an increased staining of the TCR at the surface of the WKO T cells, which also tells us that maybe these increased TCR populations will help in transmitting the activation signals. So if we want to summarize what I already said, so in our WKO T cells, we have more cholesterol in the membrane. We have an increased lipid storage. We have an alterations of the metabolisms, and this leads to, decreased T cell populations and even a higher decrease in the CD8 plus T cell populations, with higher proportions of central and effector memory T cells and an increased expression of the TCR. So we tend to have inflammatory landscapes, and this leads to the questions like, are the WQ T cells in a kind of primed state, meaning pre-activated states? So to look at the activations, we use several, how to say that? So to study the activation, we use classical WQ or wild-type mice, or mice that we crossed with an OT1 background that will allow us to activate them using antigen-presenting cells, like in a more physiological way of activations. And we recover T cells from the spleens and from the lymph nodes, isolate them, and then put them on anti-CD3 or on antigen-presenting cells for OT1 T cells. And then we look at several events of activations. And the first event during TCR activations is the phosphorylations of several activation molecules. And what we see is that we have, or in the WQ T cells, we tend to have an increase in phosphorylations of molecules such as CD3 zeta, as you can see here, meaning that we have an increased phosphorylations of these actors of the signaling cascades. And what is interesting is that we already have an increased phosphorylations of CD3 zeta even without activation, so just at the resting states. Also, let's A, for example, in that signaling cascade, seems to have an altered dynamics with an increased phosphorylations at 1 and 5 minutes, but a decreased phosphorylations at 10 minutes. So these alterations in the lipid distributions increased tyrosine phosphorylations in the early activation. And if we go a bit later, like after 5 to 15 minutes, we can use at the after 5 or 15 minutes of activations, we can use at the condensations molecules of the immune synapse. Indeed, in the T cell activations, when you have the activation signals, T cells will cluster several proteins, and the cytoskeletons will undergo complete reorganizations, and that will lead to the construction of the immune synapses with, for instance, a clustering of the TCR and an exclusion of CD45. And to look at the immune synapses, so we seeded T cells on antigen-presenting cells for 15 minutes, and we fixed, stained, and looked at the microscope. And we obtained this kind of imaging where here you can see two beautiful T cells and a clustering of the TCR on an APC that is not stained, so that you can't see. And when we analyze these immune synapses, we don't see changes in terms of number of immune synapses or area of that synapses. But when we looked at the intensity of stainings, we see that we have more or an increased intensity of the TCR staining, and also an increased intensity of the phosphozeta staining in the WQ T cells. So this means that in the immunized synapses, we have more TCR and more of that kind of activated form, meaning the phosphorylation form that will transmit the activation signals. And activations also lead to the release of the calcium within the cytoplasm of the T cells. And to look at the calcium signaling, we loaded the T cells, or like OT1 T cells, with a calcium indicator, and again put them on antigen-presenting cells, and looked at the dynamics of, or looked at the how do you say that? The intensity, or well, when we put the T cells on the antigen-presenting cells, when they activate, they release calcium that will start to blink in the contact of the calcium indicator, and will record the intensity of the T cells in a long times. And what we see is that when we put the T cells on antigen-presenting cells, we have an increased fluorescence amplitude, an increased fluorescence increase in the KO T cells compared to the wild-type T cells, meaning that we have an increased calcium release within the cytoplasm, and that's whatever the peptide dose that we used to load the antigen-presenting cells. So we have an increased calcium signaling within KO T cells. So that was for the proximal events of activations. But we can also look at later events like the cytokine secretions after 24 hours of activations. And we see an increase in the cytokine secretions like IL-6 and interferon gamma by the KO T cells. But we can also look later in activation, for example, like at the expression of activation markers at the surface of KO T cells after 24, 48, and 72 hours. And what we see is that here are the data for 48 hours of activations, but that's kind of the same for whatever the time point we looked at. And we see an increase of the activation molecules on the surface of KO T cells against whatever the affinity of the peptide we use and whatever the dose of the peptide we use. So again, if we want to sum up, in our KO T cells, we have an increased phosphorylations of the activation cascade and increased calcium signaling. This leads to increased cytokine secretions and increased sensitivity to the peptides. So our QT KO T cells seems to not be that cute anymore. And this also leads to the questions like, what are the molecular mechanisms resulting in that stronger activation of the KO T cells? So, so as I already said, activation of T cells starts with the engagement of the T cell receptors. And the behavior of the T cell receptors is known to depend on the lateral and transversal organizations of the plasma membrane. And now that we know that the membrane is disorganized in KO T cells, we looked at the diffusions of the TCR along the plasma membrane using spot variations fluorescence correlation spectroscopy. So basically, we just tracked how long labeled TCR needs to cross an elimination spots of various diameters. During that time, we'll record the photons and using correlations over times, this gives us the time for this gives us the diffusion coefficients, and this also gives us these kind of lines. And these help us, these lines, or the slope and the intersections with the y-axis of the lines characterize three diffusion regimes. When the intersections with the y-axis is 0, we have 3 diffusion regimes. When the intersection is negative, we tend to have a meshwork diffusion regimes. It means that the molecules are kind of guided by a meshwork. Most of the times, actin cytoskeletons is cited as a meshwork, and this will guide and restrict the diffusions of the molecules. And last diffusion regimes is when we have a positive intersections with the y-axis, where we have a kind of trap and domain diffusion regimes. It means that the proteins at the surface will diffuse in a way, and then will be trapped in kind of a domain and could be slowed or accelerated, and then will escape that domain, and so on. So we have 3 diffusion regimes that we can detect with that spot variations fluorescence correlation spectroscopy. And we applied that techniques on our KO and wild-type T cells. So first, in wild-type T cells, we see that the TCR diffusions at the resting states follow the traps and meshwork diffusions. But in our KO T cells, again, at the resting states, we see so I'm sorry, normally the line is a bit more down and cross here. So I don't know what happened. So in the KO T cells, what we see is that we have a meshwork diffusion regimes. It means that something starts. So we have a mesh with different regimes. And what we see also is that the diffusion is slowed down, as we see by the steeper slope of the straight lines. And what is really striking is that when we looked at the diffusions of just wild-type TCR on antigen-presenting cells that are not loaded with any peptide, we see diffusions that really looks like the wild-type ones. Here, the TCR, the diffusion of the TCR on the wild-type T cells. But when we put the OT1 T cells on antigen-presenting cells loaded with peptides, then we have a straight line that really looked like the one of the double KO T cells. So it really raised the question of, is the TCR in the double KO T cells in a kind of pre-activated conformations? And that's one of the questions that we are trying to answer during my PhD. But what is also interesting is that the double KO introduced new sensitivities of the diffusions. I mean, for example, when we treat with PP2, that will inhibit the SAC kinases, and that will then inhibit the phosphorylations of the TCR and the transmission of the activation signals. Then in the wild-type T cells, we don't see a change in the diffusion regimes, but in the double KO T cells, we kind of restore the trap and domain diffusion regimes. And when we treat with dataclin A to inhibit or to disturb the cytoskeleton organizations, then again, in the wild-type T cells, we don't see much differences, but in the double KO T cells, we end up with free diffusions. So this suggests that more lipid introduces new constraints between the TCR activation membrane and cytoskeletons. And this also raised the question, like, what this interaction between the TCR and this disturbed interaction between the TCR in the WQ T cells and the cytoskeletons, is it a clue of a disturbed actin cytoskeletons? Like, how is the actin cytoskeletons under the membrane of the WQ T cells? Is it altered by the switch in the lipid compositions? And good reporters of the interactions between the cytoskeletons and the membranes are the early adhesion and spreading. And with the great help of Rémi Taureau, previous PhD students of the LAE, we used interference microscopy to track the adhesion over time. And segmentations and tracking have been performed in a cell detective, a small software that has been developed by Rémi. And what we see is that when we put our double KO T cells on an activating surface, like meaning on anti-CD3, we see a greater spreading of the double KO T cells compared to the wild-type T cells. But when we block the SAC kinases with PP2, so when we block the phosphorylations of the TCR and of the signaling cascades, or when we put the T cells on non-activating substrates like polylysine or anti-CD45, then we don't see differences anymore. And another way of probing the cytoskeletons is to look at the mechanical properties, meaning the elastic properties. And for that, we used atomic force microscopy. So we use a cantilever with a small bit sticked on that to press on the T cells only on the first, to press on the T cells. And then we consider only the first 200 nanometers between we consider only the first 200 nanometers for analysis. So we are sure that we probe the membrane and the actin cytoskeletons without touching the cortex. And using atomic force microscopy, we end up with similar results than with interference microscopy. Meaning, when we put our T cells on non-activating substrates, like polylysine or anti-CD45, we don't see much differences between wild type and WQ T cells. But when we put the WQ T cells on anti-CD3, then the WQ T cells become softer than the wild type T cells. And when we block the transmission of the activation signals using PP2, we kind of reduce the difference between the wild type T cells and the WQ T cells. So, again, small conclusions. We see that in the WQ T cells, we have an impact on the cytoskeletons. Like first, on the TCR, we see a slower diffusions, and it starts to depend on the cytoskeleton organizations. We have an increased spreading of the WQ T cells on the anti-CD3 and a softening of the WQ T cells on that same substrates. So this raises a question on what's the real link between lipid cytoskeletons and activations, and this will need a greater investigation in the future. But if I come back a bit previously to that FCS results, so these changes in diffusions and these changes in what drive the TCR diffusions also raise the question of okay, interactions of the TCR with its environment might change because of the change of the environment, but could it also change because of changes in the conformation of the TCR? And to look at conformational changes of the TCR, we can use fluorescence lifetime imaging coupled with faster resonance energy transfer, so FLIM-FRET. So basically, we will use the lifetime of dyes, meaning how long a dye needs. How to say that? So once a dye has been excited and is in an excited state, it will require some time to come back to a non-excited state. This time is the lifetime of the dye. And when you put another dye that can accept kind of the energy of the first dye, then when you have a proximity between that donor and that acceptors, you have an energy transfer between the donor and the acceptors, and you see a reduction of the time the donor needs to come back to a non-excited state. So you see a diminution, you see a decrease in the lifetime of the donors. And these properties can be used to investigate the proximity between objects. And for instance, in our cases, we use these properties to investigate the proximity between cytoplasmic tails of the CD3 domains of the TCR and the membrane. So we loaded the membrane of T cells with R18, like a derivative of the rhodamines, and we use TCR that bear a GFP at the end of the zeta chains. We use a TCR that's that's their GFP at the end of the CD3 zeta chains. And if the CD3 zeta chains are in close proximity with the membrane, we will have an energy transfer between the GFP and the R18 in the membrane. And this will lead to reductions in the lifetime of the GFP. On the other hand, if we don't have this kind of proximity, then we don't have energy transfer and we don't see any reductions of the lifetime of the GFP. So we don't see any FLIT. So we applied that on our WQ and wild type T cells. So these are illustrations of other cells, but what interests us is on the wild type T cells, on the wild type and the WQ T cells. So when we looked first, without any acceptors, we see that we don't have differences in terms of lifetime of the GFP. That's great, that's our control. When we add R18 that will come into the membrane, then we see a decrease in the lifetime of the GFP. So we have energy transfer. It means that we have a proximity between the membrane and the CD3 zeta chains. But what is interesting is that when we compare the lifetime of the wild-type T cells in blue and of the GFP in the wild-type T cells in blue and the WKO T cells in orange, so we see that the lifetime in the WKO T cells after additions of R18 is still bigger than the lifetime in wild-type T cells, meaning that we have less energy transfer between the GFP in the wild-type T cells, in the WKO T cells than in the wild-type T cells, meaning that we have more distance between the CD3 chains of the in the WKO T cells compared to in the wild-type T cells. So this is a preliminary experiment, and we need to repeat that experiment, but these are really encouraging results that makes us think that in the WKO T cells, because of the alterations of the lipids, the organization of the plasma membrane, we have like the cytoplasmic tails of CD3 are more released in the cytoplasm and might be more accessible for the phosphorylation. So, as a big conclusions, in our WKO T cells, we have more lipid storage, more cholesterol in the plasma membrane, disorganization of the metabolism. This leads to alterations in the TCR diffusions and probably conformational changes. And this might allow an increased TCR phosphorylations and T cell activations. So that's great, but maybe these could be useful in a translational field. For instance, looking at cancers, it has been shown that alteration of the metabolism in T cells might help or decrease the T cell response against cancer cells. So we decided to look at how our T cells respond against tumor cells. So we did that in collaborations with the laboratory of Grégory Verdet in Lausanne, in Switzerland. And what we did was to take wild-type or WKO T cells. Oops, sorry, battery problem. So we took wild-type and WKO T cells, injected colon carcinoma tumor cells, and looked at the tumor growth during 2 weeks. And after 2 weeks, we collected the lymph nodes and the tumor to analyze the T cell populations that we see within the tumors. And what we see is that we tend to have a decrease in the exhausted populations. So exhaustion is a state of the T cells where the T cells are here, they are present, they are not dead, but they don't produce cytokines anymore, they don't induce cell death of cancer cells anymore. So it's a way for the cancer cells to escape the immune response. And what we see is that the WKO T cells tends to be more resistant towards exhaustions, as we see with a decreased expression of the PD-1 and a decreased expression of the exhaustion markers. In the other hand, the WKO T cells are more able to proliferate within the tumors and within the lymph nodes, and they keep higher secretions of cytokines, meaning that the lipidic organization, the lipidic alterations in our WKO T cells might make them useful tools with a greater response against tumor cells. And the first question was, why do they resist more against exhaustions, and why are they still able to proliferate? So first we have everything that I showed you previously with TCR, phosphorylations, and so on. But maybe an answer also is within the metabolism of that cells. So I already told you we have a redistribution of the phospholipids and fatty acids to neutral lipids. So we have a loss of the accessibility of that typical chains of polyunsaturated fatty acids that are not in the membrane or that are less present in the membrane of our double KO T cells. And in wild-type T cells, normally these polyunsaturated fatty acids are sensitive to and available to ferroptosis, meaning a cell death driven by oxidations of that kind of lipids. And a hypothesis was that in our double KO T cells, because these polyunsaturated fatty acid chains are less available for ferroptosis, then our double KO T cells might be less sensitive to ferroptosis. Important information is also that in tumors, ferroptosis inductions is a way for cancer cells to lower the T cell response against them. So ferroptosis inductions by cancer cells to T cells is a way of tumor growth. Let's say that. So we check the ferroptosis sensitivity in our double KO T cells, and when we tried to induce ferroptosis using so yes, when we induced ferroptosis, when we treated with the ferroptosis inductors like RSL3, we see that we only have a tiny bit of cell death in double KO T cells compared to wild-type T cells. And when we restore and we try to save the ferroptosis, then we don't see much differences. So it means that we indeed, or our double KO T cells are indeed less sensitive to ferroptosis, and this might be a first answer in why are the double KO T cells better resistance toward exhaustions. Yes, then I will finish like that. I thank you for your attention, and I'll be happy to answer your questions.
Thank you so much, Marie. Very impressive work. You have a lot of data. Thank you to give us this beautiful talk. I will read the questions we have in Zoom. One of them is, can you explain with more clarity how you define the domains in which the cell mesh network is arranged?
Well, I guess the person can't precise like okay, no, okay, I understand. Well, here I guess it refers to this kind of explanation, like when we have trap and domain regimes in spot variations for transcription spectroscopy. So the membrane of T cell is really heterogeneous, and it has been shown in plenty of studies that you can have aggregations of proteins, but also of lipids. For example, the association between cholesterol and sphingomyelins drive the agglomerations of these dimers of cholesterol and sphingomyelins, and this makes this creates kind of segregations in terms of lipids, and you can find in the literature the concept of lipid rafts, for example. So you see heterogeneous areas at the surface of the T cells with, for example, more phospholipids at some point, free cholesterol in other parts, association between cholesterol and sphingomyelins in other parts. So this is what I mean with domains, and this will also lead to the segregation of proteins with agglomerations of proteins in or outside of that domains. And yeah, so what I refer to when I say domains is that these heterogeneous areas in terms of compositions at the surface of or along the membrane of cells.
Okay, thanks. Some other questions. Your film thread data suggests a conformational change of CD3 in the DKO T cells. Do you think this reflects a pre-activated state of the TCR or rather a difference in membrane organization?
So we have several hypotheses in the sense that these film thread studies indicate us that the cytoplasmic tails of the CD3 might be more free in the cytoplasm, meaning if I come back to the first, meaning that I'll come back to the first. Gonna be easier, yes. So the film thread suggests that the tails in the wild type T cells, in the double KO T cells, might be more in the cytoplasm compared to in the wild type T cells. It means that the items here might be more available for phosphorylations. So this is an hypothesis that we have, is that these conformational changes lead to the hypothesis is that because of the lipidic disorganization of the membrane, the cytoplasmic tails are more floating in the cytoplasm, making the items more available for phosphorylations. This could explain why we see increased phosphorylations even at the resting states. And this might also explain why we see increase of the activation of the T cells and increase or this could also explain this kind of pre-activation states that we observe. Yeah. So that was the first part of the answer. Can you maybe repeat the answer, the question, please?
Wait. Do you think this reflects a pre-activated state of the TCR or rather a difference in membrane organization?
So yeah, so for us it reflects both in terms of the disorganizations of the membrane might lead to the cytoplasmic change and create these pre-activating states.
And what do you think explains the discrepancy between normal thymic development and the clear reduction of peripheral T cells in the DKO mice?
Well, that's the big question that we have indeed. So we didn't focus too much on the thymus. Where is it? Je vais la trouver. Here. So first, so T cells, when they develop, they will undergo several phases, and they will first start with a double negative state where they don't express CD4 and CD8, and then they will express both CD4 and CD8 at the surface, and then they will express only CD4 and only CD8. And in the genetical model that we have, meaning ABCA1 flux, ABCG1 flux, CD4 CRE, the double KO will only be expressed when CD4 will start to be expressed in the T cells. So it means that the double KO will only start being not available but effective at the double positive states. So at the double positive states. So maybe we have several hypotheses. Maybe it takes time for the lipidic organizations to be disturbed enough that it will lead to a decrease in the T cell populations. So maybe in the thymus, the lipidic organizations is not enough established to induce bias in terms of number, or maybe also the niche or the environment of the thymus or the metabolism of the thymic cells kind of help the T cells to survive even in the presence of the alteration of the WKO. And maybe that the metabolism within the lymph nodes or of the spleens differs in comparison to the one in the thymus. And this new environment in the spleen or in the lymph nodes, let's say, don't preserve the T cells anymore. Yeah, and what we also see is that we have an increased cell death in the spleen and in the lymph nodes. So there is definitely something happening once the T cells reach the secondary lymphoid organs.
Okay, thanks, Ryan. And the last one, because we just have like 3 minutes. It's your data show slower TCR diffusion in the DKO cells. Do you think this is mainly caused by changes in membrane lipid composition or by increased cytoskeleton constraints?
Well, that's difficult to see what drives more these new diffusions. I would answer both. Probably these slower diffusions is in the one hand because of the membrane, and in the other hand because of the cytoskeletons, and maybe it's because of both. So first, alteration of lipids might lead to differential mechanical properties of the membrane plenty of studies reports that increasing the cholesterol leads to increased packing of the lipids, making the membrane, let's say, stiffer. So maybe these changes in terms of mechanical properties of the membranes make the TCR let's say, increase or decrease the speed of the TCR diffusions. But I would say this might not be what drives the most changes, or the biggest changes, because when we look at the diffusions of other proteins, such as Ty1 or anti-CD45, we don't see such differences in terms of diffusions. It means that lipidic packings only can't explain the diffusion changes in the WKO T cells. So maybe there is specific links between how the TCR, maybe the cytoplasmic tails of the TCR interact with the membranes. Maybe the TCR at the surface of the T cells are not, or tends to be more clustered together. Maybe that's something that we have to check, and this could lead to differential diffusions. And then maybe then probably the way the TCR, the membrane, and the actins are bound together will also could also explain why we see differences in the WKO T cells, with that bond being different or being altered in the WKO T cells.
Okay, thank you so much, Mary. We hope you can visit us soon in Mexico. And well, thanks for the talk. And I let you know if we have other questions later.
Thank you very much.
Now, our next speaker, Pierre is going to present him. It's already connected.
Go ahead, can you put on your camera or try to share your screen, please.
Hi everyone.
So
thank you so much for being with us.
Thank you
We
for
are
inviting.
transmitting on Zoom. That's why you just see us. But in Zoom, we have almost 20 persons connected.
Okay.
Okay, so I will introduce Gaurav. Gaurav is also working as a PhD student in our lab in LAE. He's the PhD student of Laurent Limousin and Keya Sengupta, who you met last year during the on-site seminar series that we organized with Tatiana and the Landsbyrd. So he will present us something very interesting that is a lot in link with the IRP program we share with the Landsbyrd, which is the interaction of immune cells with partner substrates mediated by engagers. And I will let him explain you the way the engagers are designed, the way he followed this kind of interactions. And why is it so interesting is that engagers, you can characterize them from the molecular level to the cellular response, to even the functional response. And that's something that makes the link between fundamental research and medical applications. Thank you a lot. Gaurav?
Thank you for the introduction. So, hi everyone. Thank you for inviting me. And I'm going to present my work on mechanism governing the action of new molecular engager at immune cell surface. So to begin with, let's look at how immune cell normally recognize and kill cancer cell. So here you can see that I can on my cursor. So here we have a T cell, the effector cell, and it recognizes the cancer cell through a very specific interaction, the T cell receptor, and it binds to the peptide that is presented on the MHC molecule on the surface of the cancer cell. And when this interaction is correct, the cell kills the cancer cell by releasing the granzyme and other kind of chemicals that break the cell wall and kill the cancer cell. The problem arises when the cancer cell became smart and hide its MSC molecule. So it became kind of invisible from the T cell. So even the T cell is present, it is not activated. So the solution for this is to use the bispecific antibody or T cell engager. So here on the left is an example of a bispecific engager. So as the name suggests, it can bind from one side to the T cell through the CD3 receptor, and on the other side to a cancer cell through the HER2 receptor. So HER2 is an antigen which is present on breast cancer, and it can bring the T cell closer to the target cell, which can lead to the which further lead to the cell death. So we are interested in optimizing the cytotoxicity or killing of the cancer cell by designing different engager with different biophysical property, and to understand how it kills the cancer cell. So in the last couple of years, there is like many different varieties of bispecific molecules that has been produced. Some of them is already in clinic, but the mechanism, like the underlying mechanism, how they function is poorly understood. And that's why the project is two-sided. That one, we want to understand how the bispecific molecule is interacting under the immune synapse, how it is functioning. And on the other side, can we optimize the killing of the cancer cell? So to do so, we use a reductionist approach where we replace the target cell with the glass slide or a BD slide and put the antigen of interest on the surface. The advantage of such system is that one can easily play with the density and the molecule of interest. And the second is the optics, that the kind of resolution one can get through such system is not possible in cell-cell system. And I believe like this is one, the therapeutic perspective to have a goal of killing the cancer cell better, and also at the same time to understand how the underlying mechanism is working will help to understand in the future to develop more kind of better T-cell engager. So in my project, I am interested in mediating immune response via this bispecific engager. So I put the antigen of interest on the surface and the bispecific engager or T-cell engager in the solution, and I bring the T-cell and study how the T-cell interact and spread in the presence of this engager. So here we have different kind of engager with different biophysical property. Here we are playing with the length of the bispecific engager. So here you can see this is 20 base pair, which represent the 20 base pair of the double strand DNA, and we are increasing the length of this double strand DNA, and on the end there is nanobody connected with a chemical linker, so we can increase the size of this engager and hence the distance between end to end. And we are interested by increasing this length, how it will affect the spreading of the T cell. And also the molecule are developed by Juliet Breton in CRCM, which is our collaborator and working on the project, and also interested in the cytotoxicity of this experiment. And the end goal is to relate my experiment of spreading dynamics, which is occurring in 20 to 30 minutes or even less, to a long-scale experiment of cytotoxicity, which is occurring to a range of 24 hours to 48 hours. So this is how a spreading cell look like. So the imaging technique which I am using is RICM, which is reflection interference contrast microscopy. So it is based on the interference, like the signal you are seeing is the interference signal. So what happens is the light come and strike on the glass slide and also on the cell surface, and it's the interference pattern which you see. And the darker the interference pattern means the closer the cell is to the surface. So we can record the bright field, RICM, and fluorescence. Although one of the big advantage of RICM is that you don't need to have the fluorescence molecule to image it. We can just, by looking the intensity variation, can predict many different properties of such system. So here I am recording for better statistics. I'm recording 9 to 16 different field of view and analyzing 100 plus single cells. Let's look at an example of spreading of a T cell. As you can see on the left, the cell arrive and it start to spread. So what's happening is let's imagine a T cell is coming close to the surface where the antigen of interest is present, and then the cell in the first few seconds, 10 of second to minutes, the cell scan the surface, which we call as hovering phase, and decide to spread. So this is the hovering phase in which cell take before the cell start to even spread, and then finally after some time the cell finally spread. So as you can see, even some of the cell is still in this hovering state, and some of the cell has spread. Although here it is looking like a 2D image, but actually it's very interesting because here you can see there is variation in the intensity. And this is one of the example where I show you kind of a 3D intensity plot and how it is varying. So the cell will arrive and then it start to spread. And you can see each pixel is fluctuating. So what can we extract from such kind of data? First, we can understand how long does the cell take before spreading. So which we call as T of hovering, which is defined as the difference in the time when the cell start to finally spread and the cell when the cell just arrive to the surface. We can also understand the fraction of a spread cell. As you can see in this case, there is some cell which is spread, but some cell which is still in the hovering phase. We can extract the spreading area, how much the cell has spread, and finally by analyzing the dark intensity pixel or this fluctuation of intensity, and actually how low the intensity is going, one can relate it to the how close is it to the surface. So in general, the lower the intensity or the darker the intensity is, the more close is it to the surface. Maybe I will explain it again in the later part of my slide. but just to mention it here. So after recording multiple position, the first thing we can do is to normalize the image. So here the background is close to 1, and when the cell is adhering, the intensity is going below 1. For the segmentation, we use the cell pose and customize it and train it on our data. So I'm using both bright field and RACM channel to extract the mask, and I will explain why we need both the channel to extract the mask. And then we track it for extracting different measurement. So for measurement, as I was explaining, one of the things which we are interested is in understanding the hovering time. So it's the time which cell take to decide before spreading, and we want to compare it across our different engager of different length. So the first thing we would like to know is the T of first detection, which is easy to extract from the bright field, because as you can see in the RACM channel, until the cell start to spread, it's hard to see the cell. So we use the bright field to extract this information, the T of first detection, which is easy. And the next step is to extract the T of spreading onset. As you can see in the RACM channel, when the cell start to spread, the intensity start to become darker. The pixel under the mask start to become darker than the background. We can extract it by using a function which we define as area dark. So it's the number of pixels which have intensity below 0.95, as 1 is the background and it is bit fluctuating because of the noise. So we put an intensity, we took the pixel which have intensity below 0.95. By defining this function, we can produce such kind of graph. I'm showing here for a single cell, and this is the area dark with respect to time. So the cell arrived at the 5th frame. Before that, there is no mark, so the cell arrived, and then it takes some time, which is close to few 10 of second to few minutes, depending on the different condition and different engager, and then finally it start to spread. So as you can see, T of first detection is there, and to detect the T of spreading onset, we put a threshold on the area dark. So we say when the area dark start to increase more than 200, we consider it as the spreading onset. And the reason behind this 200 is that first, in case of cell which is well spread, it is clear that we are not making an error, and we are correct that the T of spreading onset is here. And in case of cell which are not spread, so as I was explaining in the beginning, there is some cell which will not spread over our experimental time, like 15 to 20 minutes, which is there. And we want to also identify those cells which will help us to know the fraction of a spread cell spreaded on in the experiment. So here the non-spread cell is fluctuating between 0 to 200, and that's why the threshold of 200 is working for us. So once we have T of spreading onset and T of first detection, we can calculate the T of hovering for this cell, and we do it for multiple cell or different field of view, and one can extract such kind of graph. So here, these are the first result from primary T cell, and it's the hovering survival curve. So here fraction is you can imagine number of hovering cell at a particular time. So, and these are the different immune engager which we have. So just to remind, 40 base pair is the one which is the longest, and 20 and 30 is the smallest one, and 30 base pair trans is the intermediate. So you can see there is a kind of hierarchy. So with the longer engager, the cell is deciding to spread faster. So what happened is that at T equal to 0, let's imagine all the cell arrive, and then after every, as you can see, there is small step. This happen when the cell go from a hovering phase to a spread phase. So you can see in case of 40, the cell is going from hovering phase to spread cell very fast. As compared to 20, with over 15 minutes, there is only one or few spread cell. And this is at 6 nanomolar. And when we increase the concentration from 6 to 16, we observe that while increasing the concentration, the smaller molecule, smaller engager also became efficient, and it started to make a bridge between the T cell and cancer cell, and it's helped to spread faster. Although there is, as you can see, not much difference in case of 40. One can again visualize the fraction of a spread cell, which I was explaining, like there is some spread cell and some non-spread cell, which one can also, which is extracted from this data only. So at t equal to 15 minutes or t equal to 10 minutes, we can look at the number of fraction of cell that has spreaded. So one can look at 6 nanomolar, there is more spread cell in case of 40. And when we increase the concentration, the difference start to diminish between the engager. It is also same for the area max. As you can see, there is a trend, more stronger trend at 6 nanomolar. And once we start to increase the concentration, the difference start to vanish. We are interested to know why we see this effect of length on hovering survival or area or fraction of a spread cell, and to test our hypothesis. But maybe before, I would also give an overview analysis of many experiments. So here, before I was showing for one experiment, here it's for more than 5 independent experiments and 2 different batch of DNAB to check the reproducibility, and it's very robust in terms of that. So here it's the rate which is extracted from the hovering curve. So from this curve, we can plot in the log scale, and by extracting the slope from this curve, we can calculate the rate. So what is this rate? This rate is telling how fast the cell is deciding to spread. So at 6 nanomolar with increase in length, so for T is again to explain the longest engager, and one can see it with the 40 base pair, the cell is deciding to spread faster, and there is more spread cell. This is the spread fraction, so there is more spread cell in case of 40 than in case of 20 or 30. So there is this nice hierarchy. And once we start to increase the concentration, for example, here it's 6 and 16, so you see that the rate that the cell is deciding to spread faster, maybe there is more number of bridge, because of which it's easy to, it's faster for cell to decide. And if you notice carefully, then you can see that in case of 40, the increase is not so much as compared to 30 and 20. So it's helping the smaller engager more to, like the increase in concentration is helping the smaller engager more to spread faster, and also to have the difference is becoming less and less when we increase the concentration. So we were interested in knowing where this difference is coming, which is the origin of this difference. And when it comes from the glycocalyx on the T cell, we know these glycocalyx are relatively long molecule, which is present on the T cell, and maybe the smaller engager were having difficulty in forming the bridge. For example, if this glycocalyx is really large, in that case, the smaller engager, like 20, which is smaller in size, can have difficulty to form the bridge. Maybe there is only one bridge, and in case of longer molecules, there can be more than one bridge. Maybe this is the reason why the smaller engager is facing difficulty. So the hypothesis was the glycocalyx impose steric hindrance, delaying cell spreading. And to understand if this is the cause, we decided to remove the glycocalyx from the cell surface by using trypsin and also by using neuraminidase in the primary cell. So here it was to check from the flow cytometry that our CD3 receptor was was intact, and we are selectively removing the glycocalyx. So here it's with trypsin, so it's the treated cell, and orange one is the untreated cell. So you can see the CD3 expression level, there is not much difference before and after treated, but there is a huge difference in CD45RA expression level. So CD45RA is the long form of the glycocalyx, like it's the long form of the CD45, and there is sugar part on the CD45, which here we can see is removed. Let's see how it affects the spreading analysis. So here it's after removing the glycocalyx, the control, the anti-CD3 was not much difference, because it was important. We were guessing maybe the treatment of trypsin or neuraminidase might damage the CD3 receptor, but that was not the case. So we were happy, and then we were looking at the different length, how it was affected. So as you can see, this is primary cell untreated, and after treating with the neuraminidase, that means after removing the glycocalyx from the surface, the cell is deciding to spread faster, and more cell is spreading, and it was reproducible with all the length. In case of 20, after treating, the cell is deciding to spread faster. In case of 40, and this was reproducible with neuraminidase and both with trypsin. So again, this was one example, and we retried it with, tried to reproduce it with different experiment and with different batch of DNAB. So here you can see this is the normal is the one which is untreated, and orange one is the one which is treated with neuraminidase, and green one is after treating with trypsin. So in all this case, one can see that after removing the glycocalyx, it's helping the cell to spread faster, and there is more number of spread cell after removing the glycocalyx. So the hypothesis that glycocalyx was hindering in forming the bridges was true. So the conclusion and summary from this part is hovering dynamics strongly depended on DNAB concentration, and as we increase the concentration, the cell start to spread faster, and the spreading area was larger. DNAB length also influence dynamics. So the longer DNAB, if you use the longer form of DNAB or engager, there was faster spreading initiation and the larger spreading area, and this difference was more pronounced at lower concentration. Experiment was reproducible, and this was tested with both primary T cell and Zucchett T cell, and it was reproduced by on many experiments. But here I am concluding that the longer molecule, the 40 base pair, is spreading faster and having large spreading area, but at the same time, the experiment which Beatrice and Juliet was simultaneously doing with using this different engager, what they found was the shorter DNAB was better in killing. So this was a bit contradiction that here I was showing that, okay, the longer molecule is behaving somehow better, that there is more spreading and more number of spread cell, but in case of cytotoxicity, why the shorter molecule is behaving better? So we were really confused, and we wanted to understand it, where this difference is coming from. And next, we put the ICAM on the surface. So till now there was only HER2 on the surface, and the bridge was mediated by only DNAB. So now after putting the ICAM on the surface, there will be two major things, that even in the absence of engager, the cell will start to adhere. Thanks to the ICAM and LFA bond, the cell will interact and the cell will spread. And the second thing will be the cell will be motile, that in the presence of ICAM, the cell will migrate. So these are the two major differences that will happen after putting the ICAM. And why we were interested in putting the ICAM was to first go more closer to the physiological condition and also to the experimental condition of the cytotoxicity. So next we will see what will happen to the metrics which I was using to understand how it will change. So the rate which I was explaining, and there was a difference in the rate before, and there was a hierarchy that the 40 base pair was spreading faster than 20 and 30. After putting ICAM on the surface, this difference diminished. And this was not a surprise because we knew that ICAM and LFA breeze is strong enough to start the spreading irrespective of the DNAM. And that's what we saw even in the absence of engagers. So no means there is no antibody, and even in absence there is sufficient spread cell, and sufficient cell is spreading at a sufficient rate. And there was not much difference in the number of cell spread. And this was repeated and validated more than 3 times. But as I was explaining, after putting ICAM, there will be a new thing that would be happening, would be cell would be moving, and so cell will be migrating in the presence of ICAM. And as you can see here, the cell arrived. And if you remember the previous video I was showing, the first video, in that case, cell was not moving. But here in the case of ICAM, cell is arriving and scanning the surface and moving. So this is only in the presence of ICAM. I didn't put any engager yet. So let's see what will happen if I put engager, DNAM. So this is at high concentration. So the cell will arrive and then stop. So this is called arrest of the cell. And here there is ICAM present. So in the case of ICAM, the cell arrive and it start to move. But if you have engager, also in presence of ICAM, the cell will arrive and stop immediately. But let's see what will happen if the DNAB is at low concentration. So you will have to wait a bit. The cell will arrive and scan a bit and then stop. If you look, for example, maybe this cell, for example, this cell, it is stopped. For example, this cell. So it's really 3 different cases that in presence of ICAM, cell arrive and it migrate. In case of ICAM and high concentration of DNAB, the cell is stopping immediately. And at low concentration, we found really interesting stuff that cell is arriving and taking some time before it's stopping. And we want to compare that in this case, and in this case, how it will vary with the different length of engager. So we can look at the velocity versus time for individual cell. So this is at high concentration of DNAB. So the cell arrive and t equal to 0 is when the cell start to spread. And as soon as it is spread, it's got arrested. So here you can see there is a bit of noise, but most of the trajectory you can see is close to 0, and it's little above to 0 because even in presence of ICAM, the cell mask is a bit moving, and that's why you get some this artifact of velocity. But you can see in case of ICAM, it's really very different. The cell is moving at a average speed of around 10 to 15, and it's not stopping. And in case of DNAB, you can see it's kind of 2 population, that there is some cell which stopped at the end, and there is some cell which is still moving. And we were interested to quantify this fraction of cell which is stopped and compare it with different, in case of different engager. To do so, we put a threshold on the intensity, which is here 3. And you can see by putting this threshold of 3, we can make all the cell in case of high concentration stopped. And in case of ICAM, by putting a threshold of 3, you can see most of the cell are up, most of the cell track are above 3. So as we see in the video, the cell are moving. And in case of DNAP, some of the cell are below 3 and some of the cell are above 3. So by choosing this threshold, we wanted to extract the number of cell which is stopped and number of cell which is not stopped. And this is the analysis for many experiment. So here you can see this is, in case of, this is x-axis is the length of the engager. So it's x-axis, it's increasing the length of the engager, and y-axis is the rate. But this time this is the stopping rate. So as we saw before, the difference in the spreading rate, like spreading onset, is not much. But here we are looking and interested in looking at the difference of stopping rate. So at a high concentration, 6 nanomolar, we don't see any difference in engager, and at concentration 0.6. So at high concentration, we don't see much difference in the engager, and even at low concentration 0.6, we don't see much difference in the engager. It's at the intermediate concentration which is interesting, that one could see that 20 base pair, in the presence of 20 base pair, cell is stopping faster than in case of 40 base pair. And this was the trend which we were very happy to get, because this was the trend of also of the cytotoxicity, that 20 base pair is kind of behaving better than 40. So here it was the first time we were able to see and kind of compare directly with the cytotoxicity experiment. And now we wanted to know why the cell is stopping faster on 20 than on 40. And here it's the effect of concentration. So it was in the spirit of producing this graph similar to the cytotoxicity experiment, where we can extract the EC50. So here again we can see the 40 is below and the 20 is above, which was matching the cytotoxicity response of the T cell. We also looked at the CD69 marker, which is known to be one of the earliest marker of the activation. So again, we looked at the RSCM channel and extract the mask from the RSM channel and look in the fluorescence channel to extract the intensity from the fluorescence channel. So here you can see this is the distribution of the CD69 signal, and we were happy to see that in case of 20, which is the smaller, there is more CD69 signal. It means it is more, in case of 20 base pair, it is more activated, and there is nice trend. Although it's this is still preliminary and we have to produce it. And this is for the granzyme analysis. So granzyme are one which will release and will kill the target cell. So here we see the control, ICAM is negative control and positive control is working well. And we can see the 20 base pair is having more granzyme as compared to 40 base pair. So this is also very, very interesting, because we can see that within 20 minutes, we can see that the cell are activated And we were guessing this stopping of the cell is related to the cytotoxicity function, because the cell, in case of 20, which is stopping early, and then it's changing the phase from a non-active to the active. So for cell to kill, it need to scan and then stop and start the functioning. So this was interesting. Next, we were also interested to see, because we were thinking in case of 20 base pair, the cell will form more close contact to the surface as compared to 40, which will really prove that when we can prove that 20 is closer to the surface than 40, we can say that because in the field, the hypothesis is the more close the cell will be to the target, the better it will be killing. So here you can see this is again a 2D representation, but here you can see the 3D representation of the intensity. And as I was explaining, the darker the pixel would be, it would mean the closer it is to the cell, which is defined by, because there is reflection first from the glass medium substrate, and second from the medium and the cell membrane surface. So this both I1 and I2 will interfere and will produce such kind of interference pattern. And as you can see, when we go below, okay, it's based on some assumptions. So this is relative, but what you can appreciate here is that once you start going below 100 nanometer, and this is the size when the cell start to adhere, and once you go below this, the darker pixel will be the more closer to the surface. But the problem with RACM signal is that because it's the intensity and height is related, but there is a cosine function, and we know that it will repeat. So the confusion would be to be sure that the intensity, for example, here at point intensity at this point might be same at this point. So there will be two different height for the same intensity. So to overcome this problem in RACM, one could use two different color, blue and green, and with two different color, one can overcome to a certain extent this ambiguity in the height, or one can use another method to understand the fluctuation in the intensity, and this is what I'm going to present. So most of my data was with the single wavelength, and I wanted to utilize this data to understand this close contact information from the RACM data. And to do so, we can imagine if this is the membrane, and if it is there is engager present there, and it is attached to the engager, then the membrane will not fluctuate much over time. But if there is no engager, the membrane will fluctuate more. So if we look at the standard deviation of last few frame when the cell is spread well, and look at the standard deviation of each pixel in last few frame, we can be sure that if there is not much fluctuation in that pixel, it is bounded by the engager. And if there is lot of fluctuation in the pixel, the engager is not present there, and maybe it's not the 40 nanometer, it's maybe 200 nanometer. So by looking at this variation in the intensity at a pixel level, one can extract the information with relative confidence that we are in the presence of engager. So what we are interested is to look if 20 is bringing T cell closer to the target than 40. So for example, in case of 20, the membrane would be here, and in case of 40, membrane will be slightly above. And to appreciate, you can already imagine this is the analysis which would reveal kind of difference in the length between 20 and 40 is not much. It's maybe relatively few nanometer, and it's really amazing that we might even able to think that one can extract such kind of information. It's like really super resolution, but in vertical. So in final, we would be expecting something like this, intensity versus number of pixels, and we would expect 20 to be slightly shifted towards left than 40. So if this will be the case, then we can say that 20 is forming more close contact to the cell. But there is a complication that there is not only engager present, there is also LFA and ICAM, and also the glycocalyx on the surface. And to reliably extract the pixel only in presence of engager is challenging, because there is not a single height. There is multiple height. And one way to deal would be to remove the glycocalyx, which I was showing that using neuraminidase and trypsin, one can remove the glycocalyx, or by not putting the ICAM on the surface. And by doing so, one can have a confidence that the pixel which we would extract will only be the pixel which is under the engager. So we can do this analysis on the last few frames when the cell is spread well. So for example, frame 85 to 90, and we can extract the mask, and we can look at the standard deviation map of last 5 pixels, and we can see that there is some pixel which is having more standard deviation, which means there is more fluctuation than other. We can look at the histogram of the intensity in the last frames, and the histogram of the STD under the mask. We can plot something like STD versus mean intensity to see how it is varying. And the goal is to put a threshold on the STD to extract the pixel which is not fluctuating much, and those will be the pixel which is bounded by the engager. So I tried different threshold on the STD. For example, this is representation of the frame, how all the pixel looks like, and the mask of the frame. And by putting a threshold on the STD, one can produce such kind of picture where this white represent the pixel which is having standard deviation less than 0.04. So these are the pixel which is really stable. And when it means really stable, means these are the pixel which is really close to the surface, and you can appreciate that we are able to reduce the pixel. For example, here one would consider all the pixel, but then it would be wrong, because some of the pixel is not bound to the engager and is fluctuating because it is the membrane. And so we tried different threshold and different threshold on the intensity, and we compare this case to look, and by extracting this pixel, we can then plot the histogram of the intensity. So here I give one example of 20 base pair to look at the histogram of intensity. And here it is without STD, so it's not it's looking like this. But when you put a threshold on the STD and the intensity, one can see that this is quite reproducible, and these are the pixels which are stable and the dark, and these are the pixel which is the pixel where the engager is present. One can this is for many different positions. One can plot something like mean of the histogram with some error bar to compare across different positions. Sorry. So the first thing was we were interested to see the effect of removing glycocalyx. So as I was explaining, we can remove the glycocalyx, and it was helping the molecule to form the bridge. And after removing, maybe it will form more bridge, and it will bring the cell closer. So one can see this is the untreated case. And after treating the case, there is reduction in the intensity, and these are the stable dark pixels. This is in case of 20. And this is in case of 40. And it is very interesting to see because, as you can imagine, in case of 20, we would expect more shift in the intensity because these were having difficulty to form the bridge. Then we also compared between 20 and 40. So this is after removing the glycocalyx. So there is only engager that is present. So we are confident that the height that we are extracting is only from the engager. And you can see that this is the 40. And there is a shift in intensity in case of 20. And this is very interesting because here it's showing that we were able to extract the difference of few nanometer. Although there is still some surprise that in presence of glycocalyx, we saw that 20 and 40 were not what we were expecting. We were expecting 20 to be more shifted to left than 40. But then again, it might be because we are not selecting the pixel, in this case, confidently from only the engager, or we are selecting some of the pixel from glycocalyx. Also, there is other complication in RSCM that once the cell come close enough, there is multiple internal reflection. For example, reflection can come from the nucleus and can spoil the intensity under the cell. So this is some of the complication which we are trying to address now, but still this is interesting. So to summarize, in presence of ICAM, the fraction of spread cell is same in case of all DNAB. So it was independent to be of the DNAB length and concentration, which was opposite in the case when we were not putting ICAM on the surface. What we saw that when we add the ICAM on the surface, there was sharp dependency on the concentration. If you notice, just by changing the concentration to 2.6, there was a huge difference that the cell was stopping or the cell were migrating. However, at the intermediate concentration of DNAB, we observed there was a wide variety of response, and we saw that with the shorter DNAB were stopping faster the cell and were having more stop cell than DNAB and of larger length. And this is interesting because it means that in case of 20, there is cell which is stopping faster, so it's becoming it's starting to function faster, and there is more stop cell. So like you can imagine, the cell need to stop to start functioning, and as soon as it stop to start functioning, it will be better. Dark pixel intensity, which is still preliminary, but maybe it suggests that 20 base pair is able to make close contact to the than the 40 base pair. And it's again interesting because but we are saying that the cell in case of 20 base pair is coming more closer to the surface, which means the CD45 protein is going out of the synapse more effectively in case of 20. And this is the kinetic segregation model, which tells that the more effectively the CD45 will go out of the immune synapse, there will be more phosphorylation, so the more signaling. So it's very interesting. And early activation marker then like CD69 and granzyme were highly expressed or more expressed in case of shorter DNAB. So if I would like to conclude and in one sentence, I would say that take-home message would be the shorter DNAB, the shorter T-cell engager promote tighter synapse formation and leading to stronger and faster T-cell activation. So it's stopping faster and it's there is more number of cell which is stopped and ready to function, and we have greater cytotoxicity function with smaller engager. With that, I would like to thank you, and I would like to thank Juliet for the molecules and Beatrice for the results of the cytotoxicity, and my supervisor, and all of you for listening. Thank you.
Thank you so much for this great seminar. We have some questions.
Yes.
So the first one, what is the main biological motivation for comparing 20 BP and 40 BP DNA engagers?
Yeah.
Is it mainly to change the physical distance between the T cells and the target surface, or?
Yeah, exactly. So maybe in the end, I was a bit explaining this point. So the understanding in the field is that the sorter molecule will be able to bring the cell more close to the surface, and then this can lead to the signaling cascade, and it will behave. It is expected to be more efficient. But even in the term of therapeutic side, it's not always advantage to have more killing, but have also to look at the cytokine release. So this was the work which Juliet did, and what they found was, although 20, for example, 20 and 30 is killing maybe in the same way, so they have same cytotoxicity, but the 30 is having less cytokine release. So you can imagine cytokine release is like doing uh, less damage to the other cell and less inflammatory response. So this height response is very, very interesting, that one can tune both the cytotoxicity response and cytokine response from this. And this will lead to the extraction of the CD45. The smaller the engager would be, it's expected that it will release more CD45 from the synapse.
Okay, and a bit related is the difference in length between 20 and 40 is only a few nanometers.
Yeah.
Do you think this small distance difference is enough to meaningfully alter receptor triggering or early signaling, or could other factors like membrane fluctuations or glycocalyx variability dominate the effect?
Sorry, I missed the full question.
It's because just to think about the difference in the really few nanometers.
Yeah, this is a bit
So
surprising, but
is difference enough, or we have like other factors like membrane fluctuations or glycocalyx variability because it's a small distance?
yeah, like as I was saying, within this small distance, we indeed saw the difference. And actually, in the field, it's known that it's not so small. Also, you can imagine the ideal distance, ideal synapse would be the 14 nanometer or 15 nanometer around that range, and around that range as the size of this engager. So it's very sensitive. It would be less effective if one engager would be 30 and 32, which would then maybe expected there is not much difference. But in this range, I think it's this small difference in length is important.
Okay, and then after, well, two more questions.
Yes.
After glycocalyx removal, both 20 and 40 BP cells appear to come closer to the surface.
Yeah.
Do you interpret this as the glycocalyx setting a minimum separation distance that overrides the effect of DNA length, or where is? I think that the question is, what is your interpretation about this, and that the two type of cells appear to come closer? Is this a minimum separation distance, or?
I think after removing after removing the glycocalyx, maybe it's more easy to answer this. So after removing the glycocalyx, we saw that the difference is easier to differentiate between 20 and 40. Like when we don't remove the glycocalyx, maybe we are not so selective in the selection of the pixel that we are only considering 20. So in presence of glycocalyx, maybe it's the distance is maybe it's still 20 base pair is forming closer contact, but maybe our analysis is not so sensitive to extract that for now. But in absence of glycocalyx and absence of ICAM, the only thing that is now making this height is this engager and the membrane fluctuation. But we have removed the pixel which is coming from the fluctuation of the membrane by using STD filter. So after removing glycocalyx, we assure that this difference is coming from the height of the engager.
Okay, and the last one is your cytotoxicity data suggests that shorter DNA base lead to better killing.
Yeah.
From a therapeutic perspective, do you think optimizing engager length could mindfully improve the performance of next generation B-specific antibodies or cell engagers?
So yeah, I missed the middle part. I understood, but can you repeat the middle part of the question?
You have your data cytotoxicity suggests that shorter as you have there in the conclusion,
Yeah.
shorter DNA ABS, better killing.
Yes.
So it's just a question of what do you think about translate this to therapeutic perspective? It means that you can optimize engagers, for example, for improve antibodies or to do other kind of cell engagers to have a better therapeutic perspective.
So what I understood, maybe I didn't understood the question completely, but yeah, the main like there's two things like the sorter, for example, here I only presented the cytotoxicity data. So 20 is killing better, but you cannot decrease the length to, like you can only decrease the length to a certain extent. There is also need for the engager to form the bridge on the both side. So there is a kind of range in which we can play. If you go below like 10 nanometer and 6 nanometer, it will be hard for engager to form a bridge. But for example, the next goal of the project is to not only vary the length, but also not the length in vertical, but also like horizontal. So we can have two different engager bind by the origami. So one can play with not just the height, but also the distance between two engager. And this is the kind of next goal to see. Yeah.
Okay, great. Thank you so much.
Thank you.
To all the speakers still connected, and of course to all people connected by YouTube. We want to just say that tomorrow and after tomorrow we will have 2 more talks, and we expect everybody being here with us to these great talks we have. Pierre.
Thank you, Goral. So, nos vemos mañana.
Nos vemos mañana.
Para el tercer día.
Sí.
Goral, thank you
Gracias.
a lot. Thank you all of us and all of you. Thank you. See you tomorrow.
See
Bye.
See you. Bye.